So we have a small crowd today, but let's start. I don't know why. It's a shame because it's a cool, very cool paper, but I don't know. I guess people were afraid from it. I'm really bad with names, so please remind me again. Jonathan, I remember, and Gabi, okay. Nissim, right. So, okay. So we'll start with the paper, with the Eric Kandel paper. Let me have it in front of me. בוקר טוב. Only the braves and the most serious students have come today. And with them we will start with the Eric Kandel paper. So let me start just by saying that compared to the other to the last paper we've read together, so this one is pretty old. It's from 1990, and correspondingly they've used pretty old essays, but still very smart essays that allows us to really, I think, decipher a lot of the details that we need in a relatively small molecular pathway, but a very significant one to neuroscience. So here maybe, because we tend to use fancy techniques today and all that, and that's good, but still it's very impressive to see what a man can do with a pair of scissors and a tape and just a few gels and come to very serious conclusions about very serious issues. So let's start as a warm-up just to go over some of the genes and molecules that are used or investigated in this paper. So what is CRE? The CRE, the CRE that is spoken about in this paper. Anyone knows what the CRE the initials, what do they stand for? Yes, Jonathan?
C-AMP responsive element.
Right, so cyclic AMP. That's what you said. So cyclic AMP responsive element. So let's start with, it's an element. It's an element on the DNA. It's a sequence, a relatively small one. So that's CRE. Let's represent it with some, so this is the DNA strand, and some sequence on the DNA is the CRE. Okay, CRE BP is what? So CRE is for the same, stands for the same thing we said for the CRE. What? So it's a binding protein. So it's CRE BP. CRE BP, it's a CRE binding protein, or cyclic AMP responsive element binding protein. That's the protein that binds on the DNA sequence CRE. So here is CRE BP. Something like that, and that will be CRE BP. Okay. And now for genes in the genome that have a CRE sequence in their promoter region, the promoter is a sequence upstream to the coding sequence of a gene. And if they have a certain sequence, the CRE sequence in their promoter region, and the CRE BP binds to it, it recruits other components of the transcriptional machinery, good morning, and then the downstream genes can be transcribed. So downstream genes would be here. Okay, and then that becomes an mRNA. Till now, we've just said that the CRE is a cyclic AMP responsive element. That's a sequence on the DNA, and CRE BP is the binding protein that binds to the CRE element on the DNA. So that's the CRE. Here, it's the sequence on the DNA, and that's the CRE BP. So cyclic AMP, as you said, it's that's what the C here comes from, and it's just the cyclic version of the good old adenosine monophosphate, and you can see it right here. So in this molecule, the phosphate, it interacts via two oxygens with the sugar ring right here, and it becomes cyclic. That's the cycle that that the cyclic AMP is named after. And now to link these three together, so upon increased levels of cyclic AMP, the CRE-BP becomes functional, and there's a cascade between these two steps that I just mentioned, and they don't really deal with it here in the paper. Today we know that the cascade involves a pKa, pKa, phosphokinase A, sorry, that phosphorylates CRE-BP, thereby causing it to become functional, and in its turn it's functional in the CRE-BP, meaning that it binds to the CRE sequence on the DNA and allows for the downstream gene to be transcribed. One more thing about the CRE-BP is that it has the motif that we've learned in the protein structure and function chapter. It has a leucine zipper, which is the binding domain, it's the binding domain, it's the motif, the structural domain that allows it to stably interact with the CRE sequence on the DNA. And what is serotonin? Sorry? It's a neurotransmitter, and you know, even today after the second hour, we'll see how it looks like. So let's wait with it to the second hour. Yeah, it's a neurotransmitter. Okay, what is long-term facilitation? Okay, so you have described facilitation or most of the time today people call it long-term potentiation. It's the exact same thing. These are the 1990s. People spoke differently, I guess. So yeah, but you described potentiation due to repetitive electrical stimulation. That's one way to induce long-term potentiation or facilitation. And what was the procedure that they have used in this paper to cause facilitation, long-term facilitation? Exposure to serotonin. So this is a kind of a chemical induction of facilitation. Okay? Most of the times, and we will even read a paper about that in a couple of weeks, to induce long-term potentiation, you inject high-frequency electrical stimulation. So here they've used the chemical version of the induction by using serotonin. And what is the molecular difference? What's the distinction of the long-term facilitation from the short one? Yeah, so the long one lasts for longer than the short one, obviously. And what is there anything that what sort of molecular process is involved in the long version in the long facilitation and does not occur? Right, so you have transcription and translation of new genes, of new protein. So synthesis of new genes and synthesis of new proteins is an essential part for the long-term facilitation process. That's what allows it or cause it to be long-term. And it does not happen in the short-term facilitation. In short-term facilitation, you might have some activation by phosphorylation of existing proteins. You might have conformational changes in existing proteins, but no synthesis of new proteins. What is gel retardation assay? Yes?
Compare regular DNA to protein-bound DNA because it will get lower in gel.
Exactly. So a molecule a DNA molecule that binds to a certain protein will be delayed on this assay, and so you will identify an interaction between the DNA and a protein molecule. Okay. And what is the electrophysiology experimental? Yes. Yeah. Sorry.
Is gel retardation assay, is that not done today? Is it an old technique?
Well, it's a general term, I think, for plenty of assays, because even okay, so I mean, even I wanted to say that electrophoresis is even you can use it as a delay assay also. But gel retardation is the way, again, the way they called it back then, and I don't think it's really, the version that they used here, I don't think it's really done today. Maybe, you know. No, I don't think so. Okay, and what is the electrophysiology experimental system? What system did they record from? Aplasia is the animal, right? But did they record from the intact animal? So where from? They've used an extract. They use a separated cells from the central nervous system of the aplasia. And as you know, and if not, Eric Kandel uses aplasia throughout his career and made, you know, miracles with it. Okay, a lot of people came at the end of this slide. I'm sorry for those who have already been here from the beginning, but just again, one thing to know, because otherwise the other slides would be meaningless. The CRE is a sequence upon the DNA. It's the cyclic AMP responsive element. Element is most usually used for a DNA sequence. CREBP is CRE binding protein. That's the green creature that binds to the CRE sequence and induce transcription of downstream gene. Okay. Okay. So that's the first. That's a gel retardation assay. Let's start with the first two lanes. What do we see in these first two lanes? And what can we conclude from it?
The first one is just the extract from the water thing.
So first one is no extract.
Yeah, the other way around. Like it's without
So what is there? So what do you have in there? It's not extract? Yeah, not this. I'm erasing this because I need the board. So picture this. You need to have it in your head. Okay. So if you don't have the extract, what do you have there? The probe of this assay. What's the probe here? What's all the black signal that you get here? Where does it come from? I mean, molecules are usually invisible, right, in a gel. So why is it black? It's a radioactive labeled something. What is the something here? What molecule is marked with a radioactive tag? The oligonucleotide. Oligonucleotides, remember, just a short sequence of nucleic acids. So what is the oligonucleotide mimicking? What's the sequence of the oligonucleotide that was used here? So in other lanes, it is indeed a somatostatin here. But let's leave it for a second. What's the probe that is so important that it is even not written here? It's the CRE. It's the cyclic AMP responsive element. So, okay. The probe all along this gel, it's the same probe and it's the CRE. It's a piece of the CRE, not a piece, just the CRE. And it's marked radioactively at the edge with some radioactive marker, and that's why we see it there. So the black cloud, that black cloud right here at the lower part of the gel, that you can see in all lanes, never mind which extract you've used or which specific. Okay, never mind. The black cloud here is the unbound probe, and the probe is the marked CRE. That's what you see in the first. So in the first line, all you see is the black cloud right here at the bottom. Okay, and now what's the second lane? What's in there? There is still no competitor, but what did we add compared with the first lane? The extract. What extract from it? It's the CNS of the aplasia. So this time we've added. Let's write the CRE right here. So this is the CRE. And then now we've added the extract, and in the extract there is some, let's call it CREBP-like protein. So that's okay, like protein. And so there's something in the extract that binds to the molecule and causes it to be delayed. That's what you see in the second lane. And more particularly, you see three distinct bands here. So there's no just one version of such an obscure, we still don't know what it is. There's no just one version of this protein in the extract, but actually there are three versions of it. And again, they didn't really go into the issue of what differentiate between the three variants of the Krebs. You just called all three bands together Krebs binding protein, Krebs binding protein like protein, Krebs like protein. Okay, so this is the first and the second lanes. So what do we need all the other lanes for? Or more specifically, let's start with let's look at the third and fourth lanes. First, what do we see in them? And second, what do they contribute in addition to the first and second lanes? Okay, so as I said, in here, the probe is still the marked Krebs. But this time they've added a somatostatin oligonucleotide. What is somatostatin? It's a name of what? Of an organism, a gene, institute of a what? Yeah, it's a gene from a rat, right? But it's a gene. Sorry? The somatostatin, is it a hormone? If it's a hormone, then it's a protein-based hormone. I'm not sure. I guess it functions as a hormone. It sounds familiar, but I'm not sure. The important thing is that it has CRE in its promoter. That's something that is already known before this paper. So we know that the somatostatin has CRE in its promoter. It has this specific element, and hence, okay, one second with the hence, and they've added the oligonucleotide of a fraction from the promoter of somatostatin, not radioactively labeled. Okay? So why do we see a decrease? Okay, this is low concentration of oligonucleotide somatostatin and high concentration. Why do we see a decrease in the intensity of the signal of the three bands when we add the unmarked somatostatin oligonucleotide? Enrica? No, okay, yes, exactly. So I mean, there's the same amount of CRE. Let's start with what you said. So it catches away or buffers out, or it's something like a titration. Actually, it titers the CREBP, because what you have now is another molecule, not radioactively marked, of the somatostatin, and the CREBP binds to it as well. And thereby it binds less to this molecule, the CRE, and more of the unbound CRE molecule would exist in this test tube and would be found. I mean, the reduction here is obviously doesn't disappear. It's more concentrated here in the cloud at the bottom. So the high concentration gives this effect more substantially than the low concentration. Everything makes sense. What do these two lanes contribute to what we have already shown here in the first two lanes? Yes, Enrica? Right. So okay. So first, it identifies what we have found in the CNS, in the aplesia CNS extraction, as something that it indeed looks like a CREBP, because that's one of the novelty in this paper. And the second is to exclude or to oppose any argument of non-specific interactions. And the reason is because, well, if you add some molecule to the extract, it might be, I don't know, maybe not just one protein, but several proteins, maybe this sequence of DNA has some features in it, and several proteins interact with it because, I don't know, because it's sort of an artifact in the gel, and it's not something that really exists in the living animal. And so this is what we call a non-specific interaction. But if you add something that you know that needs to, it's sort of a positive control, that needs to interact, or you know that it interacts with the molecule specifically, and you see that an unmarked or unmarked radioactively molecule of this thing that you know to interact with the protein reduces the intensity of the signal here. So indeed, that's what happened, and the interaction that you have concluded from the first two lanes is indeed the correct one. So I think it does two things. Again, it identifies what we have extracted from the CNS of the aplysia as something that is indeed the CREB-EP. And it opposes any argument of non-specific interactions, which is an idea that they perform again in the electrophysiology experiments. Okay, let's skip all the other lanes, which are really just different variations, different mutations, and different sources of genes with CRE in their promoters. But let's skip those let's just skip those, yes?
Is there like a way to measure the radioactivity in those single lanes? Because I think with the eye it's really hard to see. I mean, I see the effect in this one lane, but I think it's,
Yeah. Well, yeah, there are several machines and several procedures to quantify intensity in any gel assay that you have. You just, you can do it with, there are softwares to do it. You can even do it with MATLAB. You just press a line here and ask if the band's in this location, where is it strongest, or you can just mark rectangles and, counts for black pixels or whatever. Many ways to quantify it, and quite robust ones. Okay. What about this assay? So up till now, we know that the DNA, the CRE sequence interact with some proteins from the aplysia extract. And now, what does this gel assay ask? What does it answer? Maybe, but in a second, maybe someone else. Yes? Okay, yes. No, you first.
They say that if there is interaction between protein and DNA, then if you add an enzyme that cuts the DNA, it won't cut it at that region.
Right.
So it identifies the region.
Right, exactly. So, okay. Now let me repeat it with sorry, again, picture this interaction between the CRE somato and CREB-EP, and I'm really terrible with names. Please remind me your name. Adan. Okay, so what Adan said is that, first, globally speaking, so it's a gel that checks not only for interaction between a DNA and a protein, but for the specific location of the interaction between the DNA and the protein. And this is the 1990s. It's very cool. So see what happens. Okay, first, you take a DNA molecule, and there's an enzyme, as Adar said, that is called, well, you have DNase 1, DNase 2. These are enzymes, by their name, you can know that they cleave DNA, right? The as suffix indicates something that cleaves or degrades the molecule. So it's a DNase 1 that you add to the test tube together with the DNA, and it cuts it in specific let's call it sensitive points along the DNA. Say here, and here, and here, and here. So the cleavage is due to the function of the DNase 1. And what's the product of this reaction? So we would have one DNA molecule that would end here. And now an assumption we do, start with an assumption, we add the amount of an enzyme that is with a ratio of 1:1 with the amount of the DNA molecules. So most of the times, if a DNA molecule is being cut or cleaved, it will be only in one spot. There will be some instances where a single DNA molecule is cut in more than 1 point, but this would happen in extremely low frequency. So the first product would be here. The second would be this long, indicates the one that cleaved here. The third one will be this long, and the fourth, say, here. And there will be even DNA molecules that have not been cut at all. Okay. And now let's start with the fifth. Is it the fifth lane here? Yeah, the fifth lane, already for the Aplysia extract. So what are the bands here? After the cleavage, you mark these DNA molecules at the edge, again with a radioactive label. This product, which is the shortest, would be found down here in the gel. So that might be this product, right here. And the longer one will be found right above it. And following, the molecule that has not been cleaved at all would be found first right here in the first band. So do you see how this image links to what you see on the gel? Okay. And now the rightmost lane is really a ladder, what is called, or well, not really a ladder. You run only marked AG sequences, and then you get signal only in the AG spots, and you can identify the already known sequence of this piece of DNA exactly where it is. And so this is the CRE element, and this is the sequence right before and after. No, sorry. This is the CRE. The whole sequence is the CRE element, and the rectangle represents the already known binding site for the CRE element from other organisms and from other experimental systems.
How is this connected to the gel?
Okay. How is what? This one? So
What you see there, the part of it.
okay, so this gel has a resolution of a single nucleic acid. So each band here, if you would get a cleave, if a DNase would cleave the DNA in every single nucleotide, and we get band here, each band will be in parallel with a single nucleic acid.
So this is the part in the gel that is the CRE?
Yeah, this is the part in the gel that is the CRE, exactly. So that's the CRE, and that's the already known from before binding site with the CRE-BP. Okay, and now, so this was the fifth lane. And this is what people call the DNase1 footprint of the DNase1. For every enzyme, for every DNase 1, 2, or 3, coupled together, or you put it together in a test tube with a specific DNA molecule, and you will get the same footprint again and again, because it will cleave it at the same points again and again. Okay, now we perform the same thing in the fifth lane, but this time we add the extract from the first retarded band, second retarded band, and third retarded band from the previous gel. We can isolate bands from gel. You just take a pair of scissors and you cut the desired band out of the gel. You can dissolve it again and use it in other experiments. So these are the first, second, and third retarded bands. Delayed bands? Can I say retarded bands, or is it an insult? Okay, so now we perform the same thing, and this time we add the extract, which has some CRE binding protein in it, and we add it before the DNase. So the CRE-BP this time binds, let's say it binds here. Then just afterwards you add the DNase, so these 2 points will no longer be visible in the gel because the CRE-BP covers or hide these 2 cleaving points for the DNase 1. Okay? So this option is represented right here. You had a sensitive cleaving point before adding the extract, and now it's gone. Okay. But there's another option. And for that, I need another sort of sketch. Okay, so actually, when the CRABP binds to the DNA, it folds it. It causes it to bend, as we saw in previous classes, by the way. So after binding, part of the DNA molecules would be bended, say, this way. The CRABP is here. We have a binding. We have this point is still existing. This point even still exists. And there's an additional point that were not here. So here a new cutting point is formed just because of the bending here that makes it to be more sensitive to the DNA's cleavage. So the way we see it on the gel, and that has to be, by the way, outside the specific location of the interaction between the protein and the DNA, right? If there is some bending of the DNA right here inside, it doesn't matter because the protein hides it anyway. But spots nearby this protein that are being bended in some sort of way, and then and as an outcome have been exposed more to the DNA's cleavage, will be formed after adding the extract. And do you see any point on the gel that answers the description I just gave? So let's push this. Yes?
Just below the blue line?
Just below the blue line? Yeah, actually, no. This is another example of of a point that where...
No. Below the top blue line.
Below the top?
There?
But this is, again, something that existed with the DNA's... What, this one?
Yeah.
So that's the same thing as here. You had it with the DNA's one, and it disappeared after adding the CRABP. It's this option, actually. I'm pushing this button here. Above the blue line. You have, okay, instead of, so here there was there was a very faint signal right here, and it became very intensive right after adding the CRABP. And we think it's because of the structural, the conformational changes in the DNA molecule due to the interaction with the protein. Again, it has to be outside the specific site of interaction with the protein. It has to be outside it, nearby it, but outside it. It's like Sudoku. You just take, okay, that's very impressive. That's what I think. Any questions about this, gel? Okay. And now for the experimental, for the electrophysiology experimental system. So as we said, we have so we separate cells from the CNS of the aplysia, and when you grow them, so two kinds of neurons are grown in this culture: a sensory neuron that creates that form a synapse on a motor neuron, and we always record from the motor neuron. So you see that this electrode is not stick into the nucleus; it just in the cytosol, and this is the correct configuration when recording from a neuron. Okay, so the left side represents the case where we have not injected anything. That's the sort of the optimal control. And here, when you induce long-term facilitation, again, chemically by exposures. How many exposures of serotonin did they use? 5 exposures. So there, again, there are some different protocols. After 5 exposures of serotonin, there is some, different cascades, different things happen in the cell, but the outcome is that different, is that the transcription machinery is built on specific genes. Their mRNA is being transcribed. They become a protein, and things happen in the synapse. It becomes larger and becomes more active, and that's a long-lasting effect. And what do we inject here? So now you see an electrode stick into the nucleus in the sensory neuron. What did they inject into the sensory neuron? Different things, but here it's what? The what? Yeah, okay. So it's a CRE, and you have CRE inside the genome, right? So what you do here is exactly, it's an exogenous CRE, in opposed to the endogenous CRE. And what's the difference between the endogenous CRE and the exogenous CRE that they have injected? It doesn't have a gene downstream. And that's why it titers out the effect, if there will be any. Again, so this is CRE right here. And if the CRE-BP will bind to it, things will happen. The genes will be transcribed, translated, and so on. If the CRE-BP, part of it at least, will interact with the CRE that we've injected from the outside, from the external source, from the outside source, so then it wouldn't interact with the endogenous CRE, and less transcription we would have less transcription. Okay? So we inject here an exogenous CRE without the downstream gene. It's blank. Okay?
And you inject it in the sensory neuron?
In the sensory neuron. Okay, now there are some, wait 1 second, so there is some, again, today we know a lot of new things about synaptic strengthening in the presynaptic site, in the postsynaptic site, and here they inject they've performed this titration or buffering of the endogenous CRE in the sensor, in the presynaptic neuron, and that's what they say. And they record from the postsynaptic neuron, okay? And they argue things about the synapse. Okay.
So the downstream effect is the coding sequence?
Yeah, the downstream gene is the coding sequence, as I showed in the beginning, right? The coding sequence, the transcribed sequence, yeah. Okay, and let's just read this phrasing here. It's the most accurate one I could think of, and or they could think of, and it gives the reason of what exactly is the research, or what exactly is the research question in this electrophysiology part. Sorry, so if the injected CRE sequence competed for binding with the CRE-BP-like protein, it catches some of the CRE-BP. This protein might be prevented from activating the cyclic AMP-inducible genes, or bind with what it really needs to bind to, despite the increase in cyclic AMP produced by serotonin. Upon exposure of serotonin, a blockage of long-term facilitation might result. So we try to predict the outcome of this experiment due to what we think that happens, and check the results. So, okay. With no oligonucleotide, meaning just the control, yes, the wild type system, is that, so you record right before the exposure of serotonin, and you record 24 hours post the exposure of serotonin. What do we record here? It's excitatory postsynaptic potentials. You can also record field potentials from outside the cell. You can record on dendrites. But what they do here is stick the electrode inside the cell soma and record postsynaptic potentials, which in this situation are excitatory because they go towards depolarization. So they are called excitatory postsynaptic potentials. That happens due to the function of the synapse. Yeah, the signal comes from the sensory neurons and pass to the motor neurons, from which we record. Okay, so right here we've induced a long-term facilitation with serotonin, and you see this increase in the amplitude 24 hours later. So it's a long-lasting one. So this is a single case, and in a minute we'll see the quantification of n equals something. Okay, let's skip the second one for a second, and the CRE oligonucleotide. This one is that's the main player in our movie here. So when we add the CRE oligonucleotide, it doesn't allow any increase in the amplitude 24 hours later. Hence, there was no long-term facilitation. There was short-term facilitation. I don't have it here. They have it in the paper or in the supplementary material. I'm not sure, or I mention it. But there is some, in the short-term facilitation, you see some difference. You see facilitation, but not long-term one. Okay. You know what? Let's start with the quantification. They ask what did they use, why did they use the NF-kappa B and again the somatostatin. So this is no treatment. they didn't do any induction of, they didn't expose the system to serotonin. They just record a long 24 hours duration. So you see that there is some slight decrease, spontaneous deterioration of the system or something like that a day after you've started to record, because you're on the minus side. Not that significant, but something to mention. When you expose the system to serotonin and induce long-term facilitation, you see that there is an increase of 30 to 40% of bigger amplitude than was the case before the exposure. Let's jump to the last one. This is with the specific CRE oligonucleotide and exposure to serotonin, which destroys the increase in the amplitude, and hence there is no long-term facilitation. And these three are different controls for what we see here. They are controls, so it's not the heat shock, it's not the somatostatin here, but the heat shock element and the NF-kappa B element, which again are used to oppose the idea that there is any non-specific interaction in the system, meaning that if we inject the CRE and we say that it interacts with the CRE BP, which is then in its turn responsible to the long-term facilitation and so on, but it might be that the CRE sequence itself interacts with other general transcription factors in the tissue, and interacting or destroying the function of many transcription factors and showing that this one will destroys the increasing in amplitude. It's not that big a deal because you've just sort of stepped on your cells and show me that it doesn't do any facilitation. But you have to see, and they want to show something specific for the CREBP. So, and the way they showed it is that, okay, so let's inject the heat shock element, which is an element that many transcription factor binds to. Let's even do it for NF-kappa B. Again, an element that many transcription factors in the tissue bind to. And you see that there is no effect that is induced by the CRE oligonucleotide. It's only true for the oligonucleotide. And after addition of the oligonucleotide, it looks just the same as with no treatment at all. Again, no facilitation. Okay, so again, these controls were used to check for non-specific interactions. Okay, so this is what I've just said. Now it's to conclude, again, this paper has a good way to deal with criticisms about the work. So the last one I've mentioned was that, okay, maybe the long-term facilitation is affected due to non-specific reduction or titration, it's the same thing, of general transcription factors. So what they did is inject heat shock element. And then another criticism is that it might be that in the aplisma there is no really transcription factors that bind to the heat shock element, and they bothered to check this one as well using gel shifts. So the assays. So they've showed using gel shift assays that there are transcription factors that binds to heat shock element. Again, just using the delayed bands principle. So they're really using very pretty old assays. They've managed to come out with a very sound argument about a very significant pair of molecules, the CRE and the CREBP, in the aplesia experimental system. Any questions, comments about this paper?
Didn't they just show that all the processes doesn't go through the heat shock element?
It doesn't go to the heat shock. Well, that's not. I'm not sure that's the right phrasing. The long-term facilitation effect that is produced due to the activation of the CREBP does not go through the heat shock protein pathway yet. It might be the CREBP has some effect on the heat shock protein pathway, but that it doesn't have nothing to do. That it has nothing to do with the long-term facilitation in neurons. Well, they use the NF-kappa B element as well, and I don't think they've used the somatostatin in the electrophysiology experiments. But using the heat shock element, it's pretty robust. And see that there is no effect on the facilitation, it's pretty robust. It's something like using it's pretty robust. The Hitchcock element is interacts with so many genes. Okay, so my respect to the work of Eric Kandel, which, by the way, on this paper you will find Benny Hochner also, if anyone wants to ask him additional questions that I would have no idea how to answer, so he is most welcome. My respect to the Eric Kandel work is obvious after presenting this paper, but just note one slight thing. So, you know, biologists, they ask very grandiose, can I say grandiose? Okay, grandiose questions, like, is CREBP responsible for synaptic strengthening in the aplysia? And then they go and do some tiny experiment to prove their point, which is injecting the CRE element to the neuronal, to the sensory neuron and check its effect on the motor neuron on specific experimental system that is extracted from the CNS of aplysia and so on. Which is really it's an outcome of what we can do in the lab, obviously, but I think it's worth keeping in mind sometimes the gap between the experiments and the arguments the researchers do based on these experiments. Yeah, that was just a comment. Anything else? Okay, so let's take a break and continue with this with our chapter. Okay.
Potentiation is similar to facilitation?
It's the same thing. They just use the term facilitation back then, and then there's even people sometimes call it just synaptic strengthening, I think. Synaptic strengthening. Today people say LTP, LTD, long-term facilitation, latent depression.
So this was shown to be true, right? That even in mammalian systems.
Wow, in so many systems, this is the CRE, the CREBP, and the whole. Yeah, very, very true, very sound, very relevant even today in so many ways.
But you're saying it could have been just in aplysia, theoretically.
It could have been. Well, you know, yeah, it could have been. But, well, maybe not in Eric Kandel. He has so many work that is unpublished. I mean, that's what people say. I don't really know, and even Benny Hochner say so many unpublished works. So he bothers go through so many experiments until he, feel okay to publish anything about it. So in his case, I guess it's okay to believe. But today, people argue so many things based on pips. So it's sometimes a little dangerous, I think. כן. של איזה זה? כן. זה נוקלטין. כן. כן. אז יש לך רווח בגלל שה-DNA הזואן חותך רק בנקודות ספציפיות. הג'ל עצמו, אתה רואה, רזולוציה של נוקלטיד. ה-DNA לא חותך כל נוקלטיד. כן, אז אולי לא הייתי ברור שם. הג'ל עצמו יש לך רזולוציה של נוקלטיד. אם המרחק בין שתי הנקודות האלו הם 15 נוקלטידים, אז המרחק פה יהיה 15 אפס. הוא חותך את הסגנוס השלם, ואז אתה מקבל את הפרודקט. אני לא יודע, כן, אתה מן הסתם צודק, אבל...
אחרת אנחנו מתמקדים ב-A הזה.
על הדל הזה, הקונספט הוא שאתה, כאילו אתה מייצר הרבה מולקולות של זה, אתה חותך אותם בכל... אתה פשוט האמת היא מדגרט אותם לא, מה אתה עושה? אני לא זוכר שאתה מייצר אותה על הדל, האמת שאני לא זוכר. אבל לא שאתה... כן, כן, האנזים יש לו כמה מקומות שהוא חותך, ופה על הג'ל אני לא אותו, אני יכול לספור את המרחק בין הנקודה הזאת לנקודה הזאת, אני יודע את המרחק. זה יהיה 1, 2, 3, הוא חותך בכל אחד מאלה, לא? האנזים לא, הוא חותך רק פה, הוא חותך בין שני אלו. והוא חותך רק פה, הוא חותך בין שני אלו. וזהו, עכשיו פה אתה רואה שזה תמיד ה-A בולט וה-G בולט, וה-C וה-G לא אמורים להיות בולטים, אבל זה פשוט כזה סמיר על הג'ל, אז תמיד אתה מקבל בטן ב-A וב-G. ואז אתה לא יכול לטעות, הסיכוי שבטעות אתה תקבל את הפתנים האלו, אתה חופף את הפתנים ל-A ול-G ואתה יודע איפה הסיקוונס.
like this out for like the note, the review from Melissa. Right, you've seen the review next product. That's where I ran this from. Really far. So I ran down, all downhill, and so I was going backwards all the way to it.
Oh, you guys like running to the house?
No, running to like running it. And so I went down, and then as I was coming back up, I was like, this is impossible. So I like killed myself. So I got back to like 7, and then the bus started 7:20. So usually I'm fast. Like, it takes me 10 minutes. So like I just stretched and all this stuff. And so I got to the bus at like 7:25. And normally the shuttle's late. You know, it always happens like that. Whenever I'm late, the shuttle's early. So the shuttle was either early on time, and so I had to take the city bus. So it's funny because it is exhausting. You're so awake. It's like when you wake up in the morning, it's like I have like so much energy now. So I think the fact that it's going on intense. You too. What did you think of your last one? No, I'm gonna go against it. But the no, yeah, I went 6:10. No, 10. The yeah, I don't know. I have problems with my face. It's very different. And I want to sleep, and then I really want to finish at 5. But I do like that's the problem. Like you're saying, you'll do it forever, and you'll never get it. There is a gene to figure it out. Like partially it's probably a gene. How am I supposed to figure it out? How am I supposed to do what you did?
We're never given tools to do
Theoretically.
most of it. No, those are one of the things. We're supposed to do for our knowledge.
Do you get prompts to do things?
I do.
Am I supposed to find the prompt? That'll probably take me like 5 minutes. It'll probably take longer than 30 seconds. It might take me like 10 minutes.
That's the one that you're reading from. Only the smart people. If you want to be make a top like 1,000 people, like it's only we got like 10 smart guys.
It's frustrating. You think it's what? Thinking it's
Cool. It's very well styled ranking.
okay, there we go, the bus station. Antoine Pardon. Let's go.
I'm a kid, so.
Silver pen.
Really?
I have one for you,
Yeah.
but
Where'd you
I
get it?
always forget to bring it.
Amazing pen. And you describe yourself as a scribe.
Of course. What do you think? We like poor people who could do calculus in the 17th century. All these guys are rich. Lavoisier was rich, and Schurer was rich, and all these French scientists before this history of evolution.
I know, but he published actually. He published poetry against Lavoisier. He had Cleveland
Those big deals. That's not funny.
He only had 5 publications.
You look at all, every one of them. The place died in syphilis, right?
He was born in 1790.
Okay, so we will now proceed with the chapter where we stopped last time. And before that, I watched the video and noticed that I didn't give any function for the aquaporins, and there was even someone who asked a question about that here. So a word about that. So entering or exiting water molecules from the cell and into the cell. So it has, as I said, it has many functions, and I would only mention 2. The first is, well, the concentration of water molecules in the cytosol. Plenty of enzymatic reactions in the cell involve water molecules. As part of them we've met. The creation of the diester bond between nucleic acids has to do, involves the hydrolysis of a water molecule, also the peptide bond reaction, and so on. So, and many other examples for such reactions can be given. And about the tension at the membrane, so maybe the most relevant example to our course that I could think of is that, well, neuronal projections, when they are elongated during development, where, well, the terminus of the projection, what people sometimes call the growth cone, it has to have a certain membrane tension to effectively interact with certain cytoskeleton proteins from inside the cell that push the elongation towards one way or another, and this tension again is produced and maintained by exiting or entering water molecules. So these are just two functions that can be attributed to how many water you have in the cell. And then we did lots of this part, the voltage-gated ion channels. We met the potassium, sodium, and potassium voltage-gated channels. We've seen their involvement in actively propagation of action potentials along nerve cells. And okay, and now let's say, and this is where we stopped, and I want to say some I'd like to discuss some of the principal structure that these two proteins have. The sodium voltage-gated channel, which is the lower one, and the potassium voltage-gated channel. So the voltage-gated sodium channel is actually a monomeric version of the tetrameric potassium channel. So you see that each domain here should be addressed as a unit in the potassium channel, because there's just no continuity in the amino acid chain. These are four distinct units. So, so the potassium channel is a tetramer, while the sodium one is a monomer. But they have very much in common structurally wise. So for each of them, each domain or each unit is built of 6 membrane-spanning alpha helices. The 4th alpha helix is the voltage sensor because of the high concentration of positively charged amino acids in it. And that's true for the potassium and the sodium and voltage-gated calcium channels also. Between the 5th and the 6th alpha helices, we find the pore segment, the P segment. Yeah, that's the P segment. And when the four units interact with each other, the 4 pore segments are the ones that form the pore itself, form the channel through which the ion passes. And in both these proteins, there is an inactivating segment. In the sodium, the monomeric sodium voltage-gated channel, there's only one inactivation segment, while in the potassium channel we have 4, one for each, or one from each unit. See the globular domain right here. So these structural features I've described enables these proteins to be sensitive to membrane potential, open a channel, and be inactivated shortly after opening by the inactivation segment. One thing to mention is that there is high degree of homology between these 4 segments, the 4th, 5th, and 6th segments, and the non-gated channels from previous points. And that makes sense in the sense that the non-gated channels have no inactivation domain. Actually, they have homology with the 4th alpha helix, which is the voltage sensor, but it's not a voltage sensor in the non-gated channels. Okay, now a few more words about the potassium voltage-gated channel. So this is the accepted structural model for it. And the 4 globular domains that each functional channel has. So each globular domain can enter through such what we think is some kind of a lateral window right here and shut off the channel and inhibit any additional current to be transmitted through this channel. And there is a classical set of experiments that this model is based upon. Okay, so what we do here, we record currents, the potassium current. So if we do it on wild type on the intact protein, we see a robust increase in the current, in the potassium current, and then a robust decrease that is due to the inactivation. If you mutant the protein in a way that it no longer has globular domains, inactivation domains, not just for, it doesn't have any inactivation domains anymore. This is the mutant at the green. So there's a robust increase in the potassium current, as expected, but very slow or effectively really no inactivation at all, along very long duration of time. And again, something to the important experiment really here that really make this argument sound is where, because well, maybe this mutation caused, I don't know, some major conformational change in this protein, and it's no longer even resembles the wild type protein. So what they did is they add to the solution synthetic peptides that structurally mimics the inactivation globular domains. So now what you have here is a voltage-gated potassium channel with no inactivation domains, but in the solution, swimming around, you have synthetic peptides of the globular domains that are unlinked to the protein. And you again, as expected, you restore some of their inactivation feature. So you have a robust activation or opening of potassium current, and slower than the wild type, but more effective than the total mutation, and slower the activation of the potassium currents. Again, you have to imagine the domain just swimming around unlinked to the protein, and so it's an obvious intuition to why you have lower chances for the protein to be closed by these globular domains. Okay, so we've gave the mechanism by which action potentials are propagated along the axon, but for most axons in our body, well, what we described is not accurate enough, because most axons in our body are covered with the myelin sheath. And in these axons, you have regions that are covered with the myelin sheath, and in between them, gaps of exposed plasmic membrane, what is also called the nodes of Ranvier. French guy, I don't know how to pronounce Ranvier or something. Okay? So these are the nodes of Ranvier, and in these nodes, you have high concentration. This is where the voltage-gated sodium and potassium channels are really concentrated. There is no voltage channels along the covered regions of the myelin sheath. Okay, and now, so if I tell you that the voltage-gated channels are concentrated right here at this site of the plasma membrane, but I've also said at a certain time that the plasma membrane is plasmatic, it's fluidal, and proteins are constantly in constant movement along it. So how do we settle these two things together? Well, there are three known mechanisms that leads to the concentration of voltage-gated channels in these locations. The first is interaction of these channels with a family of proteins that are called, these are called NRCAM, and are a kind of adhesive proteins. These are proteins that interact very stably with proteins from inside the cell and are so localized, the exoplasmic part are pretty much localized at a certain spot. Also, the glial cells secrete that are nearby glial cells in the close proximity of the nodes of Ranvier, and recall the myelin sheath is the plasma membrane of a glial cell. So obviously you have plenty of glial cells around here. So they secrete protein hormones that trigger the clustering at the nodes. Some hormones are secreted right here in this location, and not in other locations along the axon. And they, through several interactions, it just cause clustering of these voltage-gated channels at these sites. And the last thing we know that play a part here is the tight junctions, which is something like molecular stitches between the plasma membrane of the neuron or the axon and that of the glial cell, the myelin sheath. So tight junctions, again, stitch these two membranes, the myelin sheath membrane and the axon membrane, together right here in what is called the paranodal junction, which is right here where the axon meets, or another covered region starts. This paranodal junction, where you find high concentration of tight junctions. And these tight junctions form an isolated sort of island of plasmatic membrane where there is no exchange of proteins from inside this island and with outside of this island. And so then along this region, the action potential is not really actively propagated, neither does it reduce it substantially because of the very effective isolation here. So most of the current moves axially, and then when it reaches a place with high concentration of voltage-gated channels, this is where it gets another boost. It really reforms in a way, and it gets a boost until the next node of Van Leeuwen. Yes? Because so they're not really maybe cause high concentration, you just maintain it. So if you synthesize and localize this voltage gated right here, and then you sort of stitch this area together and limit it and bound it with something, with the tight junctions, then it will hold it right here. It will hold these channels at this location, inhibit their diffusion to other locations. Okay. So that was all about the voltage-gated ion channels. And now the last part, some principal components at the synapse. So first, neurotransmitter receptors. The receptor proteins that bind neurotransmitters fall into two broad classes. The first are ligand-gated ion channels, which are themselves a channel. They recognize a neurotransmitter, a ligand, and then a channel is opened on the same protein. And the second class is what we call G protein coupled receptors, and a specific chapter, a whole chapter will be dedicated to this class of proteins which induce opening and closure of a separate ion channel. And also this is a family, a class of proteins that have much slower kinetics, and we will see why when we will get to it. But today let's focus on the first class. So, and before that, what are neurotransmitters? So most neurotransmitters we found in the cell are really amino acids and different derivatives of amino acids. Some exceptions exist, cyclic or nucleotides that lack a phosphate groups, as the cyclic AMP we've just met in the paper, can function as a neurotransmitter. Acetylcholine is again an example for an exception that's not an amino acid, it's just a substance that is synthesized in the cytosol. In some cases, short peptides can function as neurotransmitters also. And just a comment here, I think there is some disagreement between researchers to what we shall call neurotransmitters and what shall we call neuromodulators. So if you ever hear the term, and you will, so neuromodulator is a kind of a neurotransmitter that is secreted more widely, most of the time affecting more than a single synapse, about several proximate synapses. And another thing is most of the time it not necessarily activates the synapse, but really changes its sensitivity or its gain. So that's neuromodulator. Okay, let's see some of, or all of the examples I've brought here. So glycine, just the amino acid itself can function as a neurotransmitter. So same thing for glutamate. Dopamine, norepinephrine, and epinephrine are three different derivatives, or the amino acid tyrosine. Serotonin, the neurotransmitter we've just discussed, is a derivative of is derived from tryptophan. Histamine is derived from histidine, and GABA, which neurotransmitter, the favorite neurotransmitter on Hagai Bergman is derived from glutamate. So which researcher has its favorite neurotransmitter? Just choose yours. Okay.
So, with this nitric oxide, is a neuromodulator?
Nitric oxide.
Nitric acid.
I'm not sure. I'm not sure. So why do you say that? Where is?
Right. It can go through the cell membrane without.
Right. But the question, if we should call it a neuromodulator, is also the function that it induces. Or what is the outcome? So what's the outcome of nitric oxide on neurons? I'm not sure either. I don't know. I can check it out. Okay. Principally, each neuron generally produces just one type of neurotransmitter. And usually neurotransmitters are called after neurons are called after the neurotransmitters they synthesize. So when people say cholinergic neurons, they usually mean neurons that synthesize choline or acetylcholine. GABAergic neurons are neurons that synthesize GABA, and so on. Okay, and the neurotransmitters are synthesized in the cytosol of neurons and are then imported into membrane-bound synaptic vesicles and transported to the axon terminus. Vesicles are creatures of about 40 to 50 nanometer in diameter. They have a acidic lumen, which is produced by the activation, by operation of a V-class proton pump that you already know its structure and function. And this gradient of proton that is achieved by the V-class proton pump is then used to pack neurotransmitters into the vesicle by the operation of proton-linked antiporters. It looks a bit weird in first glance, I think. Use ATP to produce a proton gradient inside vesicles, and then use this gradient to pack neurotransmitters inside the vesicles. Why instead not use ATP-consuming process directly to pack the neurotransmitter into the vesicle? It's a much more efficient, rapid process. And when biologists see some not the most effective process, they raise the flag of evolution most of the time. So what we think is that the first mechanism was developed at a certain time in the evolution, and then the second one was built, was based on this. On the first one was used the mechanism that was already existing in the cell. Okay. So we'll continue with the example of the acetylcholine. But this example actually represents something more principle about neurotransmitters, packing, synthesis, and so on. But first, specifically in the case of acetylcholine, as I said, it's a molecule that's being synthesized in the cytosol from two reactants, acetyl-CoA and choline, and is then which were joined together to form acetylcholine. And this reaction is promoted by an enzyme called choline acetyltransferase. Also, acetylcholine has an antiporter that pack it inside vesicles, and it is called the proton acetylcholine antiporter. You see it right here. It works on the membrane of vesicles. Now, the gene of this protein is entirely included inside the gene encodes for this protein in the genome. So when the first protein is synthesized, meaning transcribed, the second one is transcribed as well. And this coupling makes a lot of sense if you think of it. Because if the cell needs more acetylcholine-packed vesicles, and you will synthesize only this enzyme, then this process will be a bottleneck, will limit the effective accumulation of acetylcholine inside vesicles. And vice versa, if you will synthesize this protein, you will cause higher levels of this protein, but this one and this one will not keep with the rate of production of this product. Then again, it will even be meaningless to increase the levels of this protein. So only when you increase the levels of both these proteins do we get an effective high amount of acetylcholine-packed vesicles. So in the acetylcholine case, this is the process, the mechanism of coupling between these two is simply because they are almost at the same genomic location, the genes of these two proteins. And in other neurotransmitter systems, there are other mechanisms for coupling, but you will most usually find such a coupling because it makes it much more efficient, as you saw. Okay, so the neurotransmitter have been packed inside vesicle, and it is then transported towards the axon terminus. Okay, the whole cycle that's depicted here takes about 60 seconds, I think, typically. So again, the transporters walk at the membrane of vesicles to pack inside. They use the proton gradient to pack inside neurotransmitters. The vesicles are then transported towards the membrane. They are harbored or docked. They are docking on the plasma membrane at the axon terminus. These last two steps I've just mentioned are sometimes referred to as two distinct populations of vesicles. Some experimental systems can distinguish between the first and the second. You can cause tetanic stimulation of the synapse and exhaust the second population and not the first one, and so on. The point is that some experiments draw a clear line between these two populations of vesicles, and you will, researchers will talk to you about, you will hear more about that, I think, in physiology course. And then it's a common mistake to think that the depolarization upon arrival of an action potential to the axon terminus is the one that caused the fusion of the vesicle and secretion of the neurotransmitters. Depolarization, which is the action potential, when it arrives to the axon terminus, it actually causes an influx of calcium into the cell, which in its turn cause conformational changes in proteins that interact with the dock the vesicle on the plasma membrane, and then it is fused with the vesicle is fused with the plasma membrane, and the neurotransmitter is secreted. Okay, it's not the depolarization wave or something that pushes the vesicles outside. And then the next step is a reuptake of the neurotransmitters, or before the neurotransmitters, reuptake of the vesicles back towards the cytosol. It's a process that involves a protein called clathrin. It just sort of internalize the vesicles back into the cytosol. This process goes in parallel with transporting the neurotransmitter from the synaptic cleft back to the presynaptic site. And that's a process that again involves symporters in this case. It's a symporter because you have high concentration of sodium outside the cell, and this transporter uses this downhill gradient of sodium to enter the neurotransmitter, so it takes both these molecules inside, so it's a symporter. And the uncoated vesicle becomes packed with the neurotransmitter again via the function of this transporter. Okay, that's the first half of this cycle is actually very similar to a process named exocytosis in other cells. It exists not only in neurons, but in any kind of cell. But which goes the other way around as endocytosis, which we've mentioned. And but neurons have specific features in their exocytosis process, which I will say in 1 second. Yes?
Do you pee with the clathrin is?
It's responsible to bud for the budding inside of the vesicle back towards the cytosol. It's the one that really pulls it back towards the cytosol. Okay, so the features that distinguish exocytosis in neurons compared with other cells is, first, that the secretion is tightly coupled with the arrival of an action potential. That's unique for neurons. And second is that after the secretion, you have a reuptake of the neurotransmitter and or reuptake, reforming of the vesicles. Again, I would say it like this: the local recycling of the vesicles and the secreted substance is unique for neurons. Okay, so the neurotransmitter was secreted to the synaptic cleft, and as long as it is there, it transmits a signal. And when a neurotransmitter meets its receptor, so if this is a receptor and this is a neurotransmitter, you have to think about the interaction or the binding of the neurotransmitter on the receptor as a chemical equilibrium. So the neurotransmitter binds to the receptor, and then it dissociates from it and binds again, and so on. So as long as the neurotransmitter is there at the synaptic cleft, you have transmission of a signal. And in order to terminate this signal, you just have to remove the neurotransmitter, not the receptor. It's enough to remove the neurotransmitter. And there are three ways to remove a neurotransmitter from the synaptic cleft. First, passive diffusion from the synaptic cleft. If you wait long enough, the molecule will just blow with the wind. In some cases, such as the acetylcholine case, the neurotransmitter is hydrolyzed at the synaptic cleft. And in the acetylcholine case, it is due to the functioning of acetylcholinesterase. It's an enzyme that is localized to the synaptic cleft and hydrolyzes the neurotransmitter actually very, very rapidly. So there's another enemy here that destroys the neurotransmitter, which will obviously terminate the signal. Okay, and in almost plenty of neurotransmitters, GABA and epinephrine and so on, we have transporters, as we saw in the previous slide, that reuptake the neurotransmitter back to the presynaptic site, which again would remove the neurotransmitter from the synaptic cleft. And most of them, as I've shown in the previous slides, are sodium-linked syn-porters. These syn-porters are electrogenic. We've defined this term already. They're electrogenic and hence are coupled with the transmission of a chloride ion together with the sodium back to the presynaptic site. And this process, the reuptake of neurotransmitter, and by that removal of them from the synaptic cleft, is actually at the base of many cognitive outcomes that are the outcomes of different substances. And so it's a very relevant issue in the issue of drug development, and many things, cognitive things, happens due to dysfunction or overfunction of the reuptake process. For instance, the substance cocaine inhibits dopamine reuptake. It doesn't hydrolyze it. It does not. It just destroys or disrupt with the dopamine reuptake. Fluoxetine, most familiar as Prozac, inhibits the reuptake of serotonin. And decipramine, another substance, you can look it up, inhibits reuptake of neuroepinephrine. Reuptake, reuptake is the same here. So, yes.
Do all types of neurons have all three types of removal of neurotransmitters?
Well, I would say yes in different ratios. Because even the acetylcholine, what I do know best, the acetylcholine case, after it is hydrolyzed at the synaptic cleft, I think I've even wrote it here. Yes. So it is hydrolyzed. Acetylcholine is hydrolyzed to it is synthesized from acetylcholine, but it is hydrolyzed to acetate and choline, and the choline is reuptake is being reuptake to the presynaptic side. So, one product of this reaction is reuptake, and the other, I don't know, diffuses somewhere. All three mechanisms play a role in all cells, I would say. Okay, because we've mentioned acetylcholine, so although we are more interested in the brain, but we really have to mention that this is the main, effectively the sole neurotransmitter in the periphery, in the synapse. That is not really a synapse; it's the junction between a neuron and its muscle. It's where a neuron innervates a muscle, what is called the neuromuscular junction. And so the neurotransmitter that is responsible for the signal transmission there is acetylcholine. A beautiful figure that shows the axon that innervates the muscle cell, and these vesicles you see right here are packed with with acetylcholine. Just to give some intuition of how this synapse looks like.
In another case, the lecture refers to epinephrine, sometimes as a neurotransmitter, and sometimes as a hormone.
Epinephrine, it can function as a hormone.
So some of the neurotransmitters can function as hormones too, but otherwise.
Yeah, we will define it more accurately, I think, next time. So same molecule, absolutely, same molecule can have a function at the proximate cell, at the proximate neurons, in which case it will be called paracrine signaling, and it can also be paracrine, endocrine, and faraway crine, which I will remember the term next time. And faraway crine, which is a hormone, the same molecule can function either way, and in each way each way has specific features, and we will call it differently. Yep. Okay. 2 more slides about acetylcholine receptor. Yep. So the receptor that waits for the acetylcholine at the postsynaptic site. The most common one is the nicotinic acetylcholine receptor, and it is a ligand-gated channel. Obviously, that admits it's a cation channel. It admits both potassium and sodium, and mainly sodium because of its higher gradient, but it's not specific to it. So after the neurotransmitter was secreted in the synaptic cleft, it is recognized by its receptor, the nicotinic acetylcholine receptor. In this case, it is then opened, and it delivers sodium currents, cause initial depolarization, which after that can cause a more robust depolarization, and where we will start the positive feedback that the voltage-gated sodium channels perform. This is some experiments that give some number about this release. So the release of one acetylcholine vesicle results in the opening of about 3,000 ion channels. It's not necessarily means that each vesicle here includes 3,000 neurotransmitters, because as I said, it's a chemical equilibrium between a neurotransmitter and a receptor. So another question that is not being referred to here is how long does the neurotransmitter bind to the receptor, and how fast does it dissociate from it, and so on. But okay. Was in the textbook, so okay. And for the last slide, just a couple of notes about the structure of the nicotinic acetylcholine receptor. So it is a heteropentamer. It has 5 unidentical units. Only a pair of its units is identical, the alpha subunit. And upon cooperative binding of 2 molecules of acetylcholine to this pentamer, you have some conformational change, and then what we think that happens is the following. So this is each one of these 4 is a subunit, and we just drop off one of the 5th subunit so we can look into the channel. Upon cooperative binding of 2 acetylcholine molecules to this pentameric receptor, each subunit sort of rotate on its vertical axis, and now the kink here faces towards the outside, towards the phospholipid bilayer, and this channel here, its width really increases a lot, and the channel opens. In opposed to the non-gated potassium channels and other non-gated ion channels, ions pass through this channel in their hydrated form. Again, ions pass through this channel while they are with their shell of water molecules. They do not remove it while passing, in opposed to the non-gated channels. And this high concentration of negative charges here and here, which are due to the presence of aspartate and glutamate, are thought to function as a magnet to the positively charged cations, to attract them and allow their transmission. Okay, that's a nice figure that shows that the toxic substance from a cobra venom, it is known to interact in this manner with the pentameric receptor, and by that really blocks its opening, inhibits totally its opening, and this is why when you're being where you are being bit by a snake, a cobra snake, so the first symptoms are to be are muscles paralysis, because again this is the main neurotransmitter at muscles, and it totally blocks the receptor that delivers the function of this neurotransmitter. It blocks not the binding site. It's sort of an allosteric mechanism of inhibition. It does not block the binding site. It does not even closes the channel where the ion passes, but it really interacts laterally in different sites from that of the acetylcholine, and just confirmation. It's like maybe nails that stuck this structure in this way and do not allow the rotation of the subunits. Okay, so this was a big chapter. We went over many types of ion channels and other molecules channels. For those of you who hear it the first time, please go over it again at home, and I'll be very happy to answer any question. You can send me questions by email, and I would answer to everyone anonymously. I mean, not me anonymously, but I would not say who asked the questions, so feel comfortable to ask any kind of the questions you like. Additional clarifications, and again, repeat some explanations, anything at all. That's the idea. And that's it, and we will meet next time. Okay.
line is okay, so